feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. And I'm right where I belong in my happy place. And I'm right where I belong in my happy place.
Welcome to the National University of Singapore Yongloo Lin School of Medicine's Healthy Longevity webinar. Thanks for joining us tonight. I want to remind you once again to use the Q&A function to send questions. We usually get lots of great questions. I'm sure the same thing will be true tonight because we have a wonderful speaker, Hartmut Geiger, talking about stem cells, and I'll get back to him in a few minutes. But before we do that, I want to introduce Lim Kai-shin, a research assistant in our Center for Healthy Longevity. He'll be talking about the study Evidence-Based Lifestyle Interventions for the Delay of Cognitive Decline Among Older Singaporeans, a cohort study and a randomized controlled trial.
Thank you for the kind introduction. As mentioned, my name is Kai-shin, and I'm very honored to be here to present our study that will be started in February. The study is about lifestyle interventions for the delay of cognitive decline among older Singaporeans. Currently, 1 in 10 persons above 60 in Singapore has dementia. With the increased life expectancy and a rapidly aging population in Singapore, this number is expected to increase. As there is no cure for dementia now, focus has been placed on lifestyle strategies that can prevent or delay the onset of dementia. Lifestyle factors could be classified as protective factors and risk factors. Protective factors include healthy diet, cognitive training, social engagement, and others, whereas risk factors include cigarettes and alcohol consumption, BMI, and etc. New and historic revolve that level of education, cognitive activity, BMI in late life, homocysteine, depression, stress, diabetes, head trauma, high blood pressure in midlife, and orthostatic low blood pressure are the strong predictors of dementia. Therefore, the primary aim of this study is to determine whether the lifestyle factors are associated with cognitive decline and its associated biomarkers, epigenetic age, and markers of inflammation. Moreover, we would like to see if the changes in biomarkers serve as a good predictor for the changes in cognitive functions. Also, we aim to investigate if the effects of interventions delivered in an individualized manner would produce more benefit than interventions delivered uniformly. This study combines observational and interventional study design, which use diet and healthy aging, DAHA cohort as the observational database. DAHA was formed for a protective population-based cohort study between 2011 and 2017. It collected information on both lifestyle factors and cognitive function from 1,010 community living elderly in Singapore. Then, between 2017 and 2020, a second wave of information on cognitive function was measured, along with DNA methylation and inflammation marker data. For the interventional part, the participants will be assigned to one of the three arms: uniform intervention, individualized intervention, or the control group. The control group will not receive any intervention, but the interventional group will receive group-based health education for 2 years. Within the 2 years, the health Education will be conducted weekly in group setting during the first month and monthly on the months to follow. It will consist of short talks on health-related topics that promote the lifestyle factors mentioned previously and would involve interaction to engage higher cognitive function. Participants in individualized intervention group will undergo one-on-one health coaching session for every 3 months in addition to the group session. This will primarily serve to reveal what has been taught during the group interventions and help the participants in addressing any questions and concerns they may have. The result of this study will be primarily determined by neuropsychological tests, single-digit modality tests, character tests, and stroke tests. Our secondary endpoints will be to first compare epigenetic and chronological age measured at baseline, 12 months, and 24 months through DNA methylation. Then, we will compare inflammatory markers in the blood at before and after the interventions using a 10-marker panel. We will also administer quality of life capability-related questionnaires, sleep quality and social support including ADL, IADL, EQ5D5L, ISCAP-O, PSQI, and DSSI-11. In summary, our goals are to increase the frequency of having protective activities such as physical, cognitive, and social activities. We will also try to increase the compliance of participants to change in a healthy diet, increase their sleeping quality, and maintain their mental health. Further, we also aim to decrease the frequency of smoking and alcohol consumption. Before I wrap up the presentation, I would like to call for participation from you guys or someone you know that they are eligible for the listed condition. Do reach out us at the email address. Thank you for tuning in.
Thank you. So that'll be a really interesting study to hear the results of. And we're recruiting now. So if you know anyone, please let us know. I want to introduce Hartmut Geiger, who's a professor at the Institute of Molecular Medicine at the University of Ulm. His research is really about order and disorder in cells, especially in stem cells, and how that might be driving the aging process. The thought is that restoring order in aged cells will attenuate aging and sometimes may even rejuvenate cells and organisms. So we're excited to hear about that. His talk today is titled Rejuvenation of Stem Cells by Targeting the Small RhoGTPase Cdc42. Hartmut, thanks for joining us.
Thanks, Brian, for the invitation. And I'm looking forward to share our recent findings. 1 second. Again, my name is Hartmut Geiger. I'm Institute of Molecular Medicine in Ulm, Germany, but have been also a long time at Cincinnati Children's Hospital Medical Center. So the initial research started actually in Cincinnati. And what I'm going to talk about today is stem cell rejuvenation by targeting Cdc42. So that's not new to you, but I think we should always remember that we in science and research should identify molecular and cellular mechanisms of aging that cause aging-associated tissue dysfunction and diseases. And in our case, stem cell aging might be one of the underlying causes for tissue attrition diseases, cancers, in systems that rely heavily on stem cell activity in the adults, like skin and intestine, and hematopoiesis. We'll talk primarily about hematopoiesis today. If we know the mechanisms, we could develop innovative therapies for healthy aging. Very simple, but I think it's always important. So in the hematopoietic field, again, we work on, it's a stem cell field. And on top of this hierarchy, it's the stem cell hierarchy, we have this hematopoietic stem cells. And hematopoiesis is usually located in the bone marrow, as you can see. And in this bone marrow, we have a very rare population of these hematopoietic stem cells, 1 in 100,000. They self-renew there, they do multilineage differentiation by asymmetric and asymmetric division. Then they make these progenitor cells. They're more frequent, but they have this decreased self-renewal, increased differentiation potential. And then these hematopoietic stem cells make all mature blood cells And that includes all immune cells. We should not forget stem cells make B cells, T cells, myeloid cells, NK cells, and so forth. So it's not just erythrocyte and other cells, it's the whole immune system. Good. Now I also need to introduce our favorite animal. It's a small RhoGTPase called Cdc42. You probably heard about other RhoGTPases like RAS and so forth. And GTPases that are defined by what they bind and not what they do in the sense by their presence. So only when they bind GTP, another small molecule, then they can interact with effector proteins. And then in the case of CDC42 is responsible for cytoskeletal reorganization, membrane trafficking, migration, adhesion, and many more things, which I also now refer to that is order within a cell. And the amount of GTP on CDC42 is regulated by factors that exchange GDP via GTP, and also factors that help hydrolyzing the GTP so that it becomes GTP again. And this is all regulated by many ligand receptors inside and outside the cell. So CDC42 is a good integration point of a lot of information. It'll be translated in a certain level of GTP bound, and then certain decisions in the cell will be made with respect to order. Now, a long time ago, a colleague of mine figured out that CDC42 activity is elevated in multiple tissues of mice. And we do that with the analysis with a pull-down assay. Again, it's not the expression level, it's the activity of the protein. So you have an effector protein that is able to bind to the GTP-bound form of CDC42. You can pull that out of a protein lysate, and then you develop it on a regular Western blot with a simple anti-CDC42 antibody. But as you pull it out with an effector protein, you know it has been GTP bound. This is the upper band here. And then you compare it to the total amount of CDC42 in the protein lysate of cells. It's also normal anti-CDC42 Western blot. And then you normalize it to a standard gene gap DH. And what you can see is that the activity of this CDC42 is elevated in almost all tissues that we studied so far. Some more, some less, like heart is high, liver is high, spleen, kidney. And I can also tell you that all hematopoietic cells show elevated activity. Intestinal cells show intestinal stem cells show elevated activity, and so forth. So the elevated activity of CDC4 is, I think, a novel and a likely universal hallmark of aging in stem cells and also other tissues. So now I need to go one back. So we could show that if you have a genetic tool to significantly elevate CDC42 activity in young animals, these animals clearly undergo premature aging. And we could also show that stem cells that have these hematopoietic stem cells that have this elevated level already in young animals clearly show signs of aging. So we know that it's not just a marker, it's also causative for aging, this elevated CDC42 activity. And we found first in a large set of studies a long time ago that, for example, osteopontin in stroma and osteoblasts in the hematopoietic niche, or Wnt5a within stem cells, all lead to this elevated activation of CDC42 upon aging. And what we also found, which is shown on this side, this leads to disorder with respect to standard proteins and epigenetic markers in stem cells, hematopoietic stem cells. So this is one hematopoietic stem cells. Blue is DAPI, the nucleus. The green one is tubulin, and the red one is a histone modification, histone 4 acylated on lysine 16. That in young stem cells show a polar order distribution. Upon aging, this order is gone. So you have tubulin everywhere, you have the histone marker everywhere, and so forth. This is all caused by the elevated level of GTP of CDC42. And we have shown that a drug called casein which stands for CDC42 activity-specific inhibitor, can reduce the activity of CDC42 by disrupting the loading of new GTP onto CDC42. And it does not affect other small RhoGTPases, so it's very specific for CDC42. What the drug is doing, it returns, for example, this apolar situation to stem cells into a polar one, and it also changes then the epigenetic status of stem cells and the hematopoietic stem cell function. So as we publish now, it clearly rejuvenates the function of hematopoietic stem cells if the drug has been given ex vivo to stem cells and retransplanted. So that's the whole summary. Good. Then we figured out that it can also, when given to animals in vivo, for example, alter hair follicle aging. So you can clearly show that if given to animals in vivo, old animals, you have in part here regrowth of hair follicle. Aged mice don't like to grow hairs. We could also show and publish that intestinal stem cell aging is driven by changes in CDC42 activity. If you give the drug to animals in vivo, aged ones, you can restore intestinal stem cell activity under stress situations. So you also attenuate or rejuvenate aging of intestinal stem cells. And a collaborating group in Munich here has shown that casein, so if you target that, you can affect aging and stress of mesenchymal stem cells so that you can also start addressing stress and rejuvenation of the bone marrow niche and mesenchymal stem cells, also given if the drug is given in vivo. That's all published in these papers. So, good. That means CDC42 activity elevation seems to be a central driving force in aging of the mouse. And if you can attenuate or inhibit the activity with this drug, CDC42, casein, then we can restore stem cell function in aged animals to a young level on multiple types of stem cells. Now, a mouse is a mouse. Usually we're interested in humans. So we could check also the function of aged human hematopoietic stem cells and how they look like. So first of all, this is what I said. This is now bone marrow cells from aged human beings. So this is young. You see a tiny band of GTP, CDC42 GTP, and this is an aged human being. So you see a high level on it. This is now more data. We had a large cohort of young and old people. You see the age range 20 to 40 roughly, and 60 to 80. And you see clearly that, first of all, there's an increase in relative CDC42 activity upon aging. And as you anticipate from a heterogeneous human population, you see also very heterogeneous difference in this increase. The interesting part is that the more CDC42 you have activity, you seem to change also the frequency in stem cells. It's going a little bit up, as we see, for example, also in the mouse, that you have more stem cells. And we have published that's probably due to a more symmetric division of these old stem cells while they lose their function. That's the interesting one. They have more, but they reduce their function. And we could also show that this polarity part I described for the mouse stem cells, in this case for the protein CDC42, the young ones, they are polar, and the aged ones, they are apolar. I'll come to that in a minute. Now, if you give casein back to these aged stem cells, you start to repolarize them. Again, see here, only 20% are polarized. And then if you titrate back casein, you get this up to 60% are polarized. It goes almost to the level of these young stem cells. And then we did xenotransplantation experiments to also test the function of these rejuvenated aged human stem cells in a transplant setting. The transplanted setting has some limitations because it's a xenotransplantation. So some of the function of the stem cells might not be read out perfectly. But we could see is that you have, interestingly, a bit higher contribution of these aged stem cells, might be to this artificial more cell renewal that in vivo will not lead to better stem cells in a real human being, as you have shown in the mouse. But you can see that if these aged stem cells get casein, they perform again in this assay similar to young ones. And again, because the assay is not really cool, we then started to do single cell sequencing with SMART-Seq2 and hematopoietic stem cells from a young and aged human beings. It's done by Medan Mulav, a collaborator at Ulm University, and compare young and aged stem cells first, and then the ones treated with casein. So the first thing we could do is now, in contrary to the mouse, it looks like that you can easily separate the expression profile of young and aged stem cells of humans. So you can have profiles with top 5 predictors in a PCA analysis. They can clearly tell you this is an aged stem cell and this is a young stem cell, meaning from an aged person, from a young person. And this is already the data set we used after we used AI to establish it. So really good now in separating them. And the AI training has not seen any of the stem cells treated with casein. Now, this is on the right side, you see here that we have RNA expression profiles now from donor A and B. They're all elderly donors. So this is material from 70-year-old people. And you can see donor B has only aged stem cells in the analysis. Donor A, although aged, has stem cells that are clearly aged, but also has some of the young ones. But what's more important now, if you give precisely these cells, donor cells, casein, the rejuvenation aging to donor B, then you see that all of a sudden the stem cells are to 60% young and only 40% remain aged. And even in a donor A, where you have already still some young stem cells, this goes down to 70% now being young and 30% remaining aged. We have some idea why these ones remain aged. There might not be real stem cells anymore. So we can also rejuvenate on the gene expression level with the drug human hematopoietic stem cells. Good. Now we go back to mouse. If all the functions I told you about are really true, then it would be also interesting to test whether the inhibition of CDC42 GTP with the drug in animals in vivo can modify lifespan. So we injected 75-week-old animals from day 1 to day 4, just 4 days in a row with 20 milligrams per kg, and just one injection every 24 hours. Then we took later on cytokine arrays, and then we also did at day 5 a peripheral blood analysis for methylation aging, as you're familiar with that, and we also measured survival. So now this clearly shows you that animals that received casein, the drug here, had a significant increase in median lifespan and maximum lifespan, which is around 10%, but it's the higher level of interventions that are out there. And I think we're still the only ones that can give it late in life, only for their 4 days. They never see the drug again. They have a long-term memory of what they have seen and translated in lifespan extension. And you're all familiar with biological clocks. This is done work by Wolfgang Wagner. So we used a Wagner clock for chronological aging, biological aging. So in the controls, the age controls here, you see that the chronological age and the biological age determined by the methylation clock of Wolfgang are identical. Now in the animals that got treated with casein, again, 35 days after the treatment, that's roughly here, you already see that these animals, while still chronologically aged, have a much more reduced biological age by this methylation clock. So this is not a drug that is verified on the biological aging clock level to represent this extended lifespan. Good. So summary 1, murine in human stem cells aging is driven by elevated activity of CDC42. Murine in human stem cells can be rejuvenated by inhibition of CDC42 via casein. And we see immunorejuvenation come to that. Casein extends lifespan and attenuates aging in vivo and affects most likely multiple stem cells in vivo, not only hematopoietic ones. Good. So this is what I think what aging is. As we said, if you age, you start having disorder in your stem cells, measured in our case by epigenetic disorder, but we have also other tools to measure other types of disorder distribution, and this is young rejuvenation in the cell. And such a concept could be the main driving force of aging. Good. The last 10 minutes, I would quickly go on what is the consequence of aging hematopoietic stem cell, what's going wrong in hematopoiesis. So as you know, as I said, all the hematopoietic stem cells drive the immune system. There's large discussion on whether the immune system is one of the main limiting factors for healthy aging and longevity, and what we call now aging-associated immune remodeling. You know that there's an increased susceptibility to infections, responsiveness to vaccines is decreased, and you have increase of diseases and cancers. And we think that has all to do with aging of hematopoietic stem cells. And we came up with a system to test that. It's a simple immunization schedule we do in mice. So we immunize with hepatitis B core antigen because people in a DNA vaccine, because people like that in the animal. And then later on, we measure the antibody, the T cell response by by specific peptide-loaded H2K dimers, which bind on the cognate T cell receptor that would go against the hepatitis core antigen. So we can measure immunization response in this direction in the flow cytometry chart, and we measure that in that case on CD8 T cells. And what you can see is simply in young and aged animals, you immunize them. There's less immunization in aged animals. There's no surprise. Now we can measure that in animals that got be transplanted with young stem cells and transplanted with old stem cells. We could show that these make a young immune system in these young recipients, and in this situation, you have an old immune system in old recipients. And you can also see that the age of the immune system drives the immunization response. So although you have young animals here with an aged immune system, they don't respond very well to the immunization. And that allows us to test one now if you transplant these old stem cells but they have been rejuvenated with casein, in this case ex vivo, what would be the immune response? And what you can see here nicely, this is, for example, done in spleen, as shown before. You have a very small or low immune response to the vaccination in animals that got old stem cells. Now, these animals got the same stem cells, but they were rejuvenated with casein. You're back to the levels you see in young ones. And it's even more extreme in the liver, where you have really a strong immune response of these rejuvenated stem cells. And what's even more interesting is, so casein can restore immune responses by rejuvenation of hematopoietic stem cells. And now we also looked at transcription profiles of naive CD4 T cells and CD19 B cells in this transplantation situation. So these B cells and T cells have been derived from the transplanted stem cells. And the animals only got stem cells. They did not get T cells and B cells. So they have been through a long differentiation process coming from the stem cells. And what you can see now is that these very differentiated cells that come from these stem cells show expression profiles that are on average pretty close to young ones, almost perfect for the T cells and almost perfect for the B cells, but they're very distinct from the B cells that are derived, for example, from aged stem cells. But the only time they have seen the drug was before they were transplanted, the stem cells into the animals. So what we have is a very long memory effect of the drug treatment on stem cells, and likely also other cells currently think via epigenetic mechanisms. So there's a strong memory effect within the whole system for drug treatment. Good. The last part I want to talk about is that we also figured out very recently is data of a collaborator, Yi Cheng in Cincinnati, that the drug or Cdc42 activity attenuation also directly acts on Tregs and can boost T effector cells. So this is a kind of aging or exhaustion in which we then can use them for immunotherapy against cancer. So here you have a tumor volume, In this case, it's a colon cancer model system, a very established one in mice. And if they usually have a tumor model, you give them the tumor and then the tumor grows in these animals. If you treat these animals with casein, now multiple times each day or once, then the tumor is not growing that fast. That goes along with what you have. You have Tregs that become more unstable, means they express more interferon gamma. And you can have T effector cells that increase in numbers, that both you see in these animals. And other experiments showed that this difference is really due to changes in CDC42 activity. And the best part is now that you can also combine that with checkpoint inhibition, which is currently the standard therapy in some of these cancers. And you can see that you have, first of all, tumors growing when you don't do anything. Then you have casein or a CDC42 attenuator in the animals. And you clearly see that the tumor growth is strongly inhibited. It's not fully, it's strongly delayed. Similar to what you see in the standard anti-PD-1 therapy. Now, if you combine both in this model system, there's almost no tumor growth. Actually, it's a shrinking of tumor, and that's very rare also in animal models. Good. So that means we have immune rejuvenation, attenuation of immune exhaustion, via rejuvenation of stem cells, and casein, so attenuation of CDC42 activity, can also directly act on exhausted HT cells to provide and further enhance cancer immunotherapy. With that, again, I want to mention one more time, I think that from our research now angle, this is what the core root of aging is. You go from order to disorder, and the simple trick is to restore order, and that you solve a lot of problems of aging of cells. At the end, I would like to thank my whole laboratory that has been working on. That is the Cincy group, and this is the Ulm group. All of the people listed here have contributed to one or the other way in the data that this has been presented. We have a website, and we are also ready to be viewed at Instagram. A lot of funding is necessary, as you know, to run the show. We are especially proud now that we had at Ulm University the first university-based research cluster on aging, which is called Aging at Interfaces. It's a CRC. All is done in strong collaboration with now Carolina Floria in Barcelona, I Cheng in Cincinnati, Vidani Mulafia at the University of Ulm and Reinhold, and a lot of other collaborators. I, a long time ago, gave a TED talk on aging and anti-aging. So if you want to look at that, it's still entertaining. And we also have now this funneled into a startup called Mogling Bio, which is supported by Kisu to transfer casein and casein analogs into the clinic to see where the limit is in our rejuvenation approach. You also might check that. And that's my last slide. So usually I always get a question, and you might get it too, what is this all about? Longevity, what you're looking forward to. And I think if you have something looking forward to, then this all makes sense. And what I'm looking forward to, I love to ride on trains. And this is a new high-speed train track that has been built close to my hometown Ulm here. And I was really eager to see this tunnel being occupied by real trains. And I will also continue to look forward to any new high-speed train tracks that will be built in Germany. So I have something that I want to look forward to. So it all makes sense. Thank you for your attention.
Thanks. That's a very interesting talk. And you know what? I'm going to just start at the end. Do you think it's possible to extend longevity enough so that we can see a high-speed train in the United States?
That is a really good question. So there are two variables in this, how quickly we can act on longevity and how quickly somebody in the US can act on high-speed trains. I think it will fall together. Yes, I think we can do that,
I think we have a better chance with longevity than the trains, to be honest.
That could be something, but I think if everybody speeds up efforts a little bit, I think it could fall together in
All right. Well, I'll try to
maybe
make that
we
work.
can even see high-speed trains in the US. Good.
There's a lot of questions to talk about. I think maybe I'll start with, how do you get from CDC42, active CDC42 to order? I mean, it's like there's an arrow there, and I'm trying to figure out, is this a lot of different activities of the protein? I mean, you're regulating things in the nucleus, nuclear, epigenetic control, cytoskeletal things, and yeah, well, I'll start there and then I'll continue from that.
So small RhoGTPases have been known to be in general responsible for adhesion migration, but also for polarity in yeast, for example, generation of your yeast division, and in some parts also for yeast aging if you want to. So if you do that. So this is where we initially started with. They have also been involved interestingly in memory T cell polarization. So that's also something we saw before we started looking at stem cells and all of that. So I think it's a relatively logical step to start thinking, that might play a role. And we a long time ago actually started with simple adhesion assays to determine CDC42 plays a role in stem cell aging. But then if you scratch between the surface, you find all of these other, let's say, 3D organization changes that are regulated by changes in CDC42 activity. So I would call the whole 3D space an epigenetic unit, and you might be able to change that, or that's important for cell function, especially for stem cells.
Is a lot of that effect through cell division of stem cells? Go ahead.
Yes, we have shown in one publication, did not have time to talk to, that the elevated activity of CDC42 is clearly important for more symmetric division of aged stem cells. There's a long discussion on that, and there are different views on that. I think we had done a really good job because we do single cell transplants after division. So we succeeded at that in a mouse system and could clearly show that you have a reduced overall stem cell activity, but you get more stem cells out in this aged apolar situation. And the young ones, you get a clear symmetric one. And the thinking is very simple. Then you have a balance of divisions. You have more stems, you keep your stem cell pool. And the aged one, it seems to be not clear anymore. And maybe there are also some microenvironmental influence might be stronger because you're closer to either being more differentiated or not functional. So small differences in the environment can probably push here and there. So that was our take-home message.
I'm wondering if there's a connection with mTOR because, we had done some work a while ago with Henry Jasper and Tom Rando showing that in various adult stem cells, you get chronic activation of mTOR, which pushes stem cells more toward differentiation and less toward maintenance of the stem cell state. So is there a connection between activated CDC42 and mTOR function?
So we checked a little bit. The initial results were so-so, not always clear, but also not saying yes or no at the end. But we never pushed too hard because we thought that field has been already taken and we are not sure about. But after a while, I think we should go back now and integrate it more into existing knowledge on what is out there, whether our system plays a role in that system or not, or that separate lines of how you can approach that. We have not done that in great detail, and we should do more. Yes.
One of the things I was thinking and listening to your talk is so many of the geroprotectors that people study, improve the function of adult stem cells. And so I was curious if anyone has looked at activated CDC42 in the context of, I don't know, pick your favorite intervention, rapamycin, metformin, supplements, etc.
I don't think so, because the problem here is in the assay to check for activated CDC42, these pull-down assays, unfortunately very cumbersome. You need a larger N number to be absolutely sure. And we have tried over the last couple of years to make it simpler to determine this activity, and we're still failing every now and then. So I think it's a real ordeal or task if you want to establish that in your own laboratory. So again, it's not a simple Western blot in itself. So that's the thing why people don't test it. And it's also difficult to find a simple gene signature that would correlate, oh yeah, CDC42 activity is in general elevated in the cell, because we need to work hard to align that, but then it's usually very cell type specific. So I think the model system is not easily accessible in terms of technology. It keeps, I think, other laboratories from working harder. But when they start looking at it, they see all of this. So they see all the differences that we see and report them. But they have not been tested in these other systems, yes.
Maybe I could ask a more, I want to come to the drug in a minute, but maybe I could ask a more general question first. You know, a lot of research lately thinking about aging and also disease progression is around clonal hematopoiesis. So I'm wondering how aged HSCs relate to clonal hematopoiesis and whether CDC42 has some role in that process.
Okay, I need to go one step back, and we do have a lot of research efforts on let's say, defining clonal hematopoiesis.
Yeah, that may be a good place to start for the audience anyway.
Good. So, and we are a bit off what the general consensus is currently, at least on the very level of hematopoietic stem cells. I'm not talking about differentiated progenitors or early progenitors. At the very level of stem cells, we have problems to see clonal selection towards clonal hematopoiesis. That doesn't mean it doesn't exist, but it might not be driven by stem cells themselves. It could be a progenitor, early progenitor. And if you look carefully to some of the literatures, most tools now need to go to via progenitor level to determine clonal hematopoiesis. It's not on total bone marrow or on methylcellulose assays. So tools where you have to have this differentiation and expansion to get to this level. And I'm still struggling with that, but all of our data that we have would say it's probably not on the level of stem cells. So this would then render the question, what is the drug doing to this clonal hematopoiesis? A little bit not that important anymore, because if you start looking at stem cells and transplant stem cells, then you will not see huge differences. We're in the process of testing that. The problem here is now that you cannot really do it in the human situation because there's not a good model for transplanting human clonal hematopoiesis yet. So people start to think about it. We have a collaborator at Johns Hopkins that's interested in that. And in the mouse, clonal hematopoiesis has not been described to the large extent yet. So that's where we're currently a bit struggling. But for sure, we are actively working on solving that question.
Interesting. Maybe we can talk before we go to the audience. Maybe we can talk a little bit about the drug. So I'm curious, how much you can say about how you stumbled upon this drug in the first place, and also whether it has good therapeutic-like properties for an actual drug going into the clinic.
Yes. So we stumbled upon a drug that has been defined so it's a publicly available drug first, okay? And that has been stumbled upon in a what we would never do right now, called dirty screen and xenopus extract. So you have a protein soup in front of you, and it has been identified as a drug that changes actin polymerization. That was the basic assay. So you throw in everything, throw a drug in, actin polymerization in that case was inhibited. The initial group never further pursued that, because it could be a myriad of things that are going on. And our collaborator in Cincinnati figured out, that might be really acting on CDC42 and might be a really good drug. So he then brought the targets together. And the drug is a little bit nasty because we still need micromolar concentrations to work with. On the other hand, at least in the animal studies so far, there has not been a large level or great level of toxicology. So everything we look at is usually very nice in terms of these side parts. And for sure, to use it in a clinical setting, we need to further modify it first for protection reason and then also improve it here a little bit there. And this is what now the company part is doing, because in academic setting, that's very difficult to now do hardcore drug development, because this is what's currently doing. But we have really good progress in that direction now. So you can get better translational capabilities of the possibilities, applicability of the drug. And you can also, via these modifications, make it better suitable. And I guess, from what I'm understanding, we're really good track to get this going into the clinic at one point in time, sooner than later.
I think I have two more quick questions. One is, it extends mouse lifespan, but you also showed that it affects a lot of different kinds of stem cells. So do you have any sense for whether it's through HSCs or some other stem cell population?
That's a good question, and I wish I could know that, but I wish we could actually know what is really limiting lifespan, so we could say this is our prime target, and nobody has
second that thought.
Nobody has a good answer to that. I wish we okay, good. So it definitely acts directly on hematopoietic stem cells, and Carolina Florian has shown in another paper that rejuvenated stem cells can extend the lifespan of immunocompromised animals. So this is one hint. But in the normal mouse, I don't know. For sure, I wish that hematopoietic stem cells are a main player, because that's where we're coming from. But it might be a multitude of things that extend lifespan. That needs to be still determined. Yes, of course.
And one last thought was that, this drug, could it be used ex vivo? So in other words, could you isolate bone marrow, treat the cells ex vivo, and then put them back in a human clinical context?
Yes. That's a very straightforward, easy application. The problem here is that you need to run trials on bone marrow transplants, which is not the easiest one to do for a couple of technical and health reasons afterwards. But just simply doing that is straightforward. Again, this is what you anyhow have shown and done in these manuscripts. So this is technically, and there are tools to make it in a clinical setting. Yes. So the first part of treatment is easy. The readout later on in a real clinical setting is not straightforward.
I see.
For a couple of
So,
reasons.
I could ask questions all night. I think it's a really interesting topic, but I want to bring in Anderson Koo, who's a research assistant in our Center for Healthy Longevity, and he's been monitoring the questions coming from the audience. Anderson?
Thank you, Prof, for the amazing presentation. We do have a couple of questions from the audiences. I think probably you could start with this question about the mechanism that you talked about, having the disorderness in aging. So he asked, what do you think are the mechanisms driving the increasing disorder in the nucleus? And can these mechanisms explain species differences in longevity?
Okay, so I start from the end. I don't have any clue on species differences, to be honest. That would be needing a large set of experiments, because we think our data supports that at least one main driver is this elevated Cdc42 GTP activity that can be caused by multiple external and internal pathways. And that seems to be really driving disorder. So one, we need to have comparative Cdc42 GTP biology for this lifespan and changes in animals, which we don't have simply.
I see. Okay. Moving on to the other question. This is in regards to the supplementing of the casein drug. So have you ever tried to supplement casein in vivo for several causes in aged mice? If they responded so well for 4-day treatment, the logical next step would be to repeat such treatments. So what about fitness or health span tests?
Okay. Yes, we are a little bit slow on that. That's a really cool question. No, we have not done. So the initial thinking was to measure more health span. We need to come up. We have not found the right collaboration partner now that has all the tools ready, because we don't have this fancy mouse health span boxes, everything. We're trying to get collaborators that have all of these to test these questions, but would be highly interested in. And I have one idea how we can do that, but we have not done it yet.
Okay. And
Can I interrupt just one thing there? I think that another component of that question, which I heard but maybe wasn't actually in the question, was what about if you were to do sequential treatments over some period of time? Would you get an even further enhanced effect? You probably don't know the answer, but if you could wildly speculate.
So I think with intermittent, you should get each time it goes down. I think if you overdo it, there's one thing you could overdo it based on some in vitro data. We have that. So then you can also go, if you suppress it too much, that's not really beneficial. We have some in vitro data, not really in vivo data on that. But I think it should help, that every other month or every 2 months, you can start, because again, we cannot keep it forever. It will start aging again. But there should be an interval that would be, again, helpful to do that. We have never done this because, this would be a lot of large animal control group. This to control that is not simple. That is a lot of money, a lot of time, and a lot of effort. But that's definitely on the list. If I have enough time and money, I would more than welcome doing that.
I think this question, it's quite related to what you've mentioned previously about the casein. So the question is, when considering the mechanism of action of CDC42 GTP, are there any potential side effects that could arise from the administration of casein, or by suppressing the overactivated CDC42 in aging and returning it to normal, is it unnecessary to consider any particular side effects?
Mm-hmm. So let's start again from the end. Yeah, so in theory, if you just attenuate it to the level of young ones, there should not be any side effects. The main problem is that in humans, as you've seen, there's a heterogeneous level of changes upon aging. So it's probably very difficult to titrate it perfectly to that person, although there could be another thing one could do, but that's a lot of work. So in mice, it's more due to the genetic homogeneity, it's a bit easier. Okay, good. And there can be side effects, yes. So if you really lower it very dramatically, there's also data on that. Cells don't appreciate it, especially if it's gone a long term and under high proliferation. On the other hand, having said that, we also give it to young animals. And at least in the setting we had in this four every day for 4 days, there seems to be no side effect also in young animals. When you say you could even go a little bit lower, there seems to be a kickback mechanisms that goes relatively quickly back to normal. So maybe the memory part is I want to stick normal, and anything lower or higher, if you can adjust it somehow, then they try to keep this normal. That's my hypothesis. I don't have really clear data on that though.
Okay, thank you very much for that answer. We have another question about the results of the methylation clock. So, what is the difference in methylation clock if comparing the casein treatment and other treatment in mice? Do they share almost similar changing clock, or is there anything specific that's different about it?
That's my big point currently. I'm not sure that what the current status is on other methylation clocks in longevity animals after treatments. My gut feeling says we're really, really at the leading edge of what you can see with these clocks, especially that we have measured relatively early after the treatment, and we see this already 9 weeks difference. So we think we are definitely in sync with what's out there. We need to check carefully if we're the leading edge or just in the pack currently. Okay.
Yeah, I think that it's somewhere near the leading edge, I would say. I think the challenge is that everybody's using different clocks and different treatments and different ages, and it hasn't really been systematized. But, if you look at the published literature on other geroprotectors, that's certainly, a robust effect.
Mm-hmm. Yes, and it's a really good clock. It's not the Horvath clock. It's the one from Wolfgang, but that that is really, at least for the chronological correlation, it's as tight as Steve's clock. So you have 90.9 or something for a mouse part, for the Black 6 part. Good.
We have another question. So since you showed that casein increases T cell functions, can casein be used as an adjuvant to vaccines in elderlies?
This could be one future use for a drug like that. Now, from the development side, that is not an easy trial to run either. And as you know, some of the initial Restore Bio interventions for that, although I think they had a little bit difficult design at the end. So in theory, it's doable practically to run a trial to make this a clear yes or no. I've also learned it's not that easy to run due to the multiple variations have, and that it's not simple to find clear endpoints in such a trial. So you need a really large trial to have good endpoints. So it's not straightforward, but for sure, that is a application to test if you find somebody wants to run a really large trial. Good.
I think building on the future possibility of this drug, casein, there was another question about have any studies explored the efficacy of the casein drug in models of neuronal degenerative diseases?
We are in the process of doing so, and my gut feeling is that could actually be very nice results coming out of that. So because like neurons have a real big 3D transportation organization problem, as you know, they have these meter long neurites, and we know that CDC42 is important for getting things going in neurons. It's not but it's published data. So there could be some really good beneficial effects also in neurotransport disorganization problems, which I think have to do with aging. Yes, good.
Yeah, I think we are approaching the end of the Q&A. I think we probably do one last question. So I think this is not really limited to just the casein drug, but of course you can answer casein for this one. So if you had $1 billion, what would you test on humans and possibly create a new drug?
We'll give you 10 million. I don't want you to be limited by cash here.
10 million.
So 1 billion, not 10 million.
Good. So I'm a little bit self-centered currently because initially I didn't believe our data on the drug part that we have. There's a lot of things on the market. So I would really put a lot on our drug currently and our intervention because I think it has very, very solid data in multiple settings now. But definitely I would also bet a lot or invest a lot on general lifestyle changes. So what you can support by simple interventions, because pharmacological interventions have pros and cons at the end. You might always have some side effects. There are probably a lot of other interventions that are beneficial, or would also put a lot more effort into drugs like metformin that are already on the market and might have very beneficial effects if tested correctly in larger cohorts, because currently there's a problem in getting all the money going into something that is not fully protected. And I think this is where we should also put our effort into, or if you have unlimited cash, I think there's a lot to harvest at the end for having some information, which is beneficial to everyone. But again, for companies, it will not be because they cannot protect and work with that. And I think there's a large gap in this field, but I can also understand why this is. We can only solve it with free-flowing cash and large amounts of free-flowing cash.
Sounds good to me. So thanks a lot, Hartmut, for giving the talk. We'll look forward to getting an update in the future. Thanks, Anderson. You're a natural at this. You're going to take my job if I'm not careful. I want to remind everyone to use the chat function and choose the panelists and all attendees option to leave comments and feedback on the show. Please sign up for our Unlock Healthy Longevity Supplements Conference, February 29th to March 1st. It starts on Leap Day. I don't know what that means for the conference, but it'll be exciting, I think. This is actually the first global conference focused solely on the role of supplements as a geroprotector interventions. It'll have scientists, clinicians, industry partners, and consumer reps to explore the possibilities. Also, mark your calendar for Saturday, March 2nd, as we'll unveil the truth about health supplements. Wow, that's scary. During our special open day, delve into the comprehensive study exposing misleading labels on immune health dietary supplements and join experts in unraveling the science behind genuine longevity solutions. That'll be March 2nd, so please come for that. The links to all these things are inside the chat box. Our next episode will be February 8th. We have one of our very own, Dean Ho, who's the director of the Institute for Digital Medicine, or Wisdom, here at the National University. And he'll be talking about digital longevity medicine. He'll also mention, I think, his new book, which is kind of N=1 clinical studies. And so that'll be interesting. Andrea Meyer will be hosting that show. And I want to leave you with a final video on healthy lifestyle. Thanks for joining us.
It's important to remember our physical health affects our mental health and vice versa. What we do to our bodies makes a big difference to how we feel. Physical activity, diet, alcohol, smoking and drugs can all affect our mental health and well-being in different ways. So let's talk about them. Physical activity releases feel-good hormones called endorphins, which help us sleep and feel better. It also improves our physical fitness, which tends to make people feel better in general. Even small amounts of regular physical activity can improve your mental well-being, especially if it's doing something you enjoy. Physical activity is even a recommended treatment for some types of depression. You can get active at your own pace in your own way. There's no need to join a gym or even spend any money. Simple small changes in our day-to-day can make all the difference. Taking a brisk walk at lunchtime, or walking to get where we're going, getting active in the garden, or cycling once or twice a week are great options. If there's a type of activity you used to enjoy, think about how you could pick it up again. For example, there are lots of local groups and sports teams open to all levels of ability. What's more, studies show that time in green spaces is beneficial. So head to your local park if you can. If you need a little help fitting more physical activity into your day, try the Active 10 app. It helps you get short bursts of brisk walking into your day. There's also the fantastic Couch to 5K app, which will get you up and running in just 9 weeks. Eating better can play a big role in our health and well-being. It won't come as a surprise to learn that what we eat and how much plays a big part in our physical health, which can impact our mental health too. Ideally, we should aim to eat a healthy, balanced diet. Whatever you choose to do, what's good for your body is good for your mind.
I'm feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. I got my head held high, I'm on my